Hello everyone. Today's notes are on Gregor Mendel. So we briefly mentioned Gregor Mendel before. He's known as the father of genetics. He actually was not a scientist, but he was an Austrian monk, so kind of like a priest. He lived in a monastery, and he worked with pea plants. So basically he was a gardener because a lot of monasteries produce their own food, and a lot of his studies come from his experiences working with plants. His work was published in 1865, and he was not given this title of Father of Genetics until after he died. Mendel's experiment, like I said, was with pea plants, and this is why you'll see a lot of Punnett Square questions about pea plants. And the reason that he used peas was because they're available, they're easy to get, they reproduce in large numbers, so each pea plant will produce many peas, which helps with determining your data set. You're having a large sample size. They're easy to work with, easy to grow, and they produce many generations in a short time, so he doesn't have to wait a year to do another test. When we are talking about how Mendel did his experiments, it's important to be familiar with the structure of a flower, because this is how he was able to control his experiments. So in most flowers, we have a male structure and a female structure. The male structure are these parts here. They are known as stamen, which is easy to remember because it has men in it, and it produces pollen in the anther up here. And the pollen goes through meiosis to form sperm cells. So plants reproduce sexually as well. This center part here is known as a carpel or pistil. Pistil is probably the more common term used. This is the female structure, and it produces the ovule or eggs down here in the bottom. In plants, pollination is fertilization. So fertilization is when the male sex cell, the sperm, and the female sex cell, egg, joins. So just to review, what is the fancy word for a sex cell? If you said gamete, then you would be correct. So pollination is fertilization in plants. It's when the egg and pollen, which is really the sperm cell, are going to combine to make a seed. There are two types of pollination, self-pollination and cross-pollination, which I'll go through in the next few slides. What this diagram is showing here is very cool. This structure here is the top of the pistil. You can see all these bright dots. These are where the pollen would have landed because the top of the pistil is sticky. It's called the stigma, and that helps attach pollen if it's blowing around in the wind or it gets brushed onto it from an animal like a bee. The pollen granules grow a tube down towards the egg, which is what you're seeing here, and those sperm cells are going to travel down the tube where they can eventually reach the ova or egg and to combine for fertilization. Self-pollination, just like it sounds, is when a single flower can pollinate itself. So garden pea plants are self-pollinators. The pollen from a flower can pollinate the eggs from the same flower. So you see here, very simple. The pollen from the anthers will go right onto the stigma, and these sperm cells will be able to pollinate this egg cell. And that can be a good thing because it means that they don't have to worry about being close to another plant for pollination, but it can also be a bad thing because it can reduce genetic diversity. Cross-pollination is when we have pollination between two different plants. So sperm from one plant is going to fertilize an egg from a different plant. This can be done through many ways. Wind, like in the springtime when you go outside and your car is all yellow from pollen, that is an example of wind pollination. It's being spread through the wind. Insects such as bees, that pollen will stick to the hairs on their body, and then when they go and land on another flower, that pollen can brush off. And then humans can also control pollination, which is what Mendel did in his experiments, and a lot of gardeners will use this to help increase how much fruit or vegetables they get in a certain year. For Mendel's experiment, he studied one trait at a time, so that he was doing a very controlled experiment. In pea plants, each trait has two different versions, and the different versions of a gene are known as alleles. We talked about dominant and recessive alleles, such as in pea plants we have tall or short plants, round or wrinkled peas, yellow or green peas, etc. Mendel did controlled pollination. He would pollinate the plants himself, which guaranteed that only the pollen he was trying to experiment with would fertilize the egg. And the way that is done is by removing the stamen from a flower so that there's no chance of self-pollination. And then you take that pollen and you brush it onto the flower that you want to fertilize. And then that pollinated carpel will mature into a pod, which gives us the peas, and he can study their characteristics. As we mentioned in one of the other note sets, this is what Mendel's experiments often looked like. He took parents that were true breeding, meaning one was homozygous dominant, another was homozygous recessive, so we here have a tall plant and a short plant. When he crossed them together, he would get all heterozygous offspring, so we have two different alleles, versus the parents, which were homozygous, where the two alleles are the same. And then he would further cross those to see what traits the offspring had, which is how he was able to determine that many of these heterozygous plants were carrying the recessive allele. From his experiments, he came up with 3 ideas that became laws. And in science, a law is a statement that summarizes scientific facts, versus a theory, which is an accepted explanation based on experimentation. A theory can change and be adjusted, but laws are usually not. Our first law is the law of dominance, and this states that the allele expressed for a particular trait is dominant, while the other trait that is not expressed is considered recessive. So if we look at this cross between, again, 2 true breeding parents, we see that in the offspring, even though they each have a recessive allele, they're going to show the dominant trait, which is tall. Our second law is the law of segregation. And because every organism has 2 alleles for each gene, but when we go through meiosis to produce gametes, we have half the chromosome number, these alleles are going to be separated. And then they are recombined during fertilization, which gives us combinations that may be different from the parents. So even though in this case we have 2 heterozygous parents, it's possible to have offspring that show the recessive trait. The law of independent assortment is the third one, and it states that genes are independently separated from one another during meiosis. Because of crossing over when those chromosomes are exchanging information or pieces of their structure, we are going to get separation of traits. And this allows for alleles for different traits to be distributed independently and give us more variation. So really the laws are pretty simple. The law of dominance states that some traits are dominant, some are recessive, and when an organism has both, they're going to show the dominant trait. The law of segregation is that alleles are separated, and then law of independent assortment focuses on genes being separated and sorted into gametes separately from each other. Lastly, write your summary and questions in your packet and come check in with me. And I will post a video on Classroom if you would like a review.